Yeah, okay. So good evening, students. So let's begin off with what we have started off in the previous session. So if you could recollect, in the previous session, we started off a topic called as sex determination. So we have said that there are different ways of which how we determine the sex in a particular organism. And with regards to that, we saw about two different types of sex determination shown in different organisms, right? So in correspondence to that, the first type what we started off is called as the chromosomal basis of sex determination, right? So under chromosomal basis of sex determination, we have gone through that the chromosomes are actually involved in determining the sex of an organism. So as per that, we saw about something called as the male heterogamety as well as the female heterogamety in the previous session. So under that, we spoke about something called as XX and XY type of sex determination and XX and X0 type of sex determination, where the males are the ones which are actually responsible for determining the sex of a particular organism. The second category or second type what we saw was with regards to female heterogamety, and under that we took about something called as the ZZ, ZW type of sex determination as well as the ZZ and ZO type of sex determination. In this category, it is the females who are actually responsible for determining the sex of a particular individual or a particular organ. So we have taken off a couple of examples also under each of these categories what we have, or each of these types what we have taken. The second type of sex determination what we saw was in case of Drosophila, right, or in case of fruit fly, right, where the type of sex determination is based on the ratio of X chromosomes to the ratio of autosomes, right? To the ratio of X chromosomes to the autosomes. In simple, we call it as the X by A ratio. So based on the X by A ratio, the sex of an organism would be determined, whether it is a male or whether it is a female. So we have seen a few ratios also, right? If the ratio is X by A ratio is said to be 0.5, we said that it is a normal male. If it is 1, we call it as normal female. If the X by A ratio lies between 0.5 and 1, we call it as intersex or gynandromorphs, right? Where part of the body appears like male and part of the body appears like female. Then we went on into if the X by A ratio is less than 0.5, we started calling it as meta males or super males. And if the X by A ratio is more than 1, we call it as meta females or super females, where both the meta males as well as the meta females are said to be sterile organisms. Right? So in this type of sex determination, the Y chromosome does not have any role in determining the sex of an organism. Instead, I told you that Y chromosome is responsible for determining the fertility of the male organism. Right? So that is what we have seen in the previous session. So in this session, let us continue with another type of sex determination. Then we'll move on into the next topic. So the type of sex determination what we are taking up now would be termed as something called as haplodiploidy type of sex determination. It is called as haplodiploidy type of sex determination. Now, what exactly is this haplodiploidy type of sex determination? So let us get into the details of this haplodiploidy type of sex determination. Before that, this haplodiploidy type of sex determination is usually seen in a class of insects which is called as Hymenoptera. Hymenoptera, right? In class of insects called as Hymenoptera, we have this type of sex determination which is called as haplodiploidy type of sex determination. One example what we can take for this haplodiploidy type of sex determination is in case of honeybees. Right. I think all are familiar with honeybees, so there is nothing much to get into the details of this. Right. So in honeybees, the type of sex determination is called as the haplodiploidy type of sex determination. Right. So before we get into this type of sex determination, let us talk a few things about the honeybee as well as the honeycombs. So whenever we talk about honeybees, we know that there are 3 individuals present in the particular comb, what we actually call it as hive or the honeycomb. Right? There are 3 different individuals present in the honeycomb. One is, we call it as a queen, right? So this is the first individual here. The second one is called as the worker, and the third one is called as the drone. So these are the 3 individuals which constitute the family of the honeybees. right? So there is one queen, right? And remember, in a honeycomb or in a beehive, there will be only one queen. So this queen is actually a female individual, right? Workers, there are a lot of workers present in the beehive or the bee comb, and this worker is also said to be a female individual, and drones are the male individuals, right? So these are the three members of the honeybee family. So there is a queen, which is a female, worker, which is a female, and drones, which are the males, right? Now, if you see the number of chromosomes present in case of honeybees, the diploid number of chromosomes in case of honeybees is 32, whereas the haploid number is said to be 16, right? So this is the chromosome number in case of honey. Diploid number is said to be 32, and haploid number of chromosomes, it is said to be 16, right? Now, among these queen, workers, and drones, the queens are said to be diploid individuals. So queens and workers both are said to be diploid individuals, whereas the drones are said to be haploid. So this you need to remember, right? So queens and workers, queen and workers, they are said to be diploid in number. That is, they have 32 chromosomes, whereas the drones are said to be haploid. They have only one set of chromosomes, so they have only 16 chromosomes, right? So this is the basis of what we are actually going to take up. So let us talk about this haplodiploidy type of sex determination. Why we had called it as haplodiploidy type? Haplo, because the drones are said to be haploid. Diploidy type, queen and workers both are said to be diploid in nature. So that type of sex determination, we call it as haplodiploidy type of sex determination. So let us see how exactly the sex determination is done in these particular organisms, right? So let's take up a few processes as well, right? See how exactly sex determination is done in these organisms. Let's begin with the queen, right? So we said queen is said to be diploid in number, right? So the queen, which is a female, is said to be diploid in number, right? And we have drone, which is haploid in number. Now this queen, which is diploid in number, before it undergoes the process of sexual reproduction, it undergoes a process called as gametogenesis. And we know what exactly happens in gametogenesis, right? In gametogenesis, a diploid number chromosome is being reduced to a haploid number through a cell division, which is called as meiotic cell division. So we have studied about all of this in detail also, right? So this diploid queen, after undergoing a process of meiosis, will start forming two haploid gametes, right? So this is through a process called meiosis. They undergo a process called meiosis where they start forming two haploid gametes, right? Now, what about drone here? So drone is already haploid. Now, if this haploid drone has to form a haploid gamete, which type of cell division will usually take place here? It would definitely be mitosis. So this is an important one, what you have to remember. So the drones undergo mitotic cell division to form the male gamete, right? So this would be the male, right? So the queen would undergo meiotic division to form the female gametes, whereas the drones would undergo mitosis to form the male gametes, right? So why is the drone undergoing the mitotic division? What happens if it again undergoes meiotic division? We know that in meiosis, what exactly is happening? The chromosome number is being reduced to half. Now, if here already it is haploid, if again meiosis occurs here, what would be the condition? It would become n/2. That is not possible at all. So it undergoes equational division, that is mitosis, to form the male gamete. So once the male gamete and the female gametes have been formed, this female gamete will start fusing with the male gamete through the process of fertilization, right? So through the process of fertilization, they start forming a diploid zygote. A diploid zygote is being formed. Now, since the diploid zygote is being formed, what would be the sex of the diploid zygote here? Since it is diploid in number, so the sex of this individual would be female. Right. Now, how exactly is a male being formed here now again? This second gamete of the queen will now undergo a process called as parthenogenesis. Parthenogenesis. So once it undergoes the process of parthenogenesis, it would now get transformed into a male organism. So this would be male, right? So this would now get transformed into a drone, right? A drone is being formed. So through the process of parthenogenesis, the female gamete, right, containing a haploid set of chromosomes will be transformed into a drone having only one set of chromosomes, that is 16 chromosomes. Whereas the other female gamete would fertilize with the male gamete, right, and form a diploid cell, which is called as a zygote. Now, this diploid zygote would be a female, and a male drone, which is formed as a result of parthenogenesis, would be termed as a parthenogenesis would be termed as a drone. Right. So this is how the males and females are actually being formed in this particular case. Now, what would happen next again? Again, this particular zygote, right, once it becomes an adult organism, it again undergoes a process of gametogenesis, again forms a diploid cell, right? Whereas this drone again, by undergoing mitotic cell division, will form another haploid male gamete. Now, this male gamete and the female gamete would fuse together to form a diploid zygote, which is again a female, whereas this female gamete would now again undergo a process called as parthenogenesis. Again, it would be transformed into the male drone. So this is how the sex of the organism is being determined. Right? So what exactly is happening here in this particular case of haplodiploidy type of sex determination? If the individual is diploid, if the individual has two sets of chromosomes, such type of individual, we start calling it as the female individual. If there is only one set of chromosomes involved or present, such type of individual would be termed as the male individual, and that male is called as the drone. Right. Now, keeping this aside, now, when a queen and a drone mates with each other, what exactly is happening? A diploid queen would now undergo a process of meiosis, that is reduction division, and forms two haploid gametes, right, which we call it as the female gametes. Similarly, the drone, which is a male, would now undergo equational division, that is mitosis, and forms the male gamete. So the female gamete and the male gamete would be fused together through a process called fertilization, and it would lead to the formation of a diploid zygote. Now, the sex of this zygote as well as the individual will be female. Why it is a female? Because they have two sets of chromosomes. Their diploid number of chromosomes is being present. Similarly, here, the other female gamete, which is actually present here, will now undergo a process called as parthenogenesis and get transformed into a male organism, which is called as the drone. Right? So this drone, again, now during the process of reproduction, sexual reproduction, again, it starts forming haploid gametes by undergoing a process called mitosis. This female organism, that is the queen, again would start producing haploid gametes by undergoing a process called as meiotic cell division, as a result of which the male gamete and the female gamete would fuse together to form a diploid female again, whereas the haploid gamete, right, that is the female gamete, would undergo a process called as parthenogenesis again to form a male drone. So this is how sex determination is done. So once this is done, I told you that this female gamete and the male gamete would be fused through a process called fertilization and forms a diploid cell. Now, I told you that this diploid cell will be a female individual. Right. Now, we have two different types of females here. One is called queen and the other one is called workers. Right. Now, this female has, again, we have two different types here. One is called as the queen and the other one is called as the worker. So, both are again diploid individuals and both are again said to be females. Right. Now, among all the female organisms that are formed, right, among all the female organisms that are formed, one female organism would be selected among all the female progeny. Right, try to understand this. among all the female progeny, one female would be selected, and that female would be fed with something called as the royal jelly. Right? So one female would be fed with royal jelly. So the female, a diploid female, which is fed with royal jelly, will be transformed into a queen. Right? A female individual which is being fed by royal jelly would be transformed into the queen. Whereas all the other females, whatever are there, they get transformed into workers. Now this queen bee will actually be the fertile bee in the complete beehive, right? Whereas the workers are not said to be fertile, right? So this is what we have. So what exactly we have seen here? Once a diploid zygote is formed, among these female organisms, one female organism would be selected, and that female organism would be fed with royal jelly. And the female individual or organism which is fed with royal jelly would be transformed into the queen, right? And that queen will be the fertile one, right? And the major role of queen is to undergo the process of gametogenesis and undergo the process of sexual reproduction. That is the work of this queen. Whereas all the other work of the beehive or the honeycomb is done by this workers, right? And as I've already said, in a beehive or in a bee comb, right, there will be only one queen that is being present. Once this queen dies, what happens once the queen dies? Once this queen dies, this queen will start releasing certain chemical substances, right? The queen will start releasing certain chemical substances. Now, these release of this chemical substance is an indication which is given for the other workers, so that these workers again would now select one female individual and start feeding it with royal jelly. Therefore, that individual or that female organism which is fed with royal jelly will now again become the queen. Right? So that's how this particular process of sex determination is done in case of honeybees. We start calling it as haplodiploidy type of sex determination. Right. One more important thing here is, right, I told you that the female gamete would undergo the process of parthenogenesis to form the male drones. Right. So this process of male parthenogenesis, this process of male parthenogenesis is called as erhentoky. An important term here, what you need to remember, right? So this process of male parthenogenesis is called as erhentoky, which is actually shown by the members of this particular organism called housefly. Right? So this is how the sex is being determined in case of housefly, where we start calling it as haplodiploidy type of sex determination. Right? So this is the third type of sex determination what we have. After what we have taken up two types of sex determination in the previous classes. Right? One was the chromosomal basis of sex determination, where we said chromosomes are the ones which are involved in determining the sex of the organ. Second one, what we said in case of Drosophila, it was the ratio of X chromosomes to the autosomes. So the ratio of X chromosome to the autosome would determine the sex of the organ. The third one, what we are seeing now is haplodiploidy type of sex determination. Right? So whether an individual is haploid or a diploid individual, so that haploid or diploid nature would determine the sex of the particular individual. So this is what we have with haplodiploidy type of sex determination. Right. So one more important thing what you need to know here in this particular haplodiploidy type of sex determination is what most of the scientists say is the drones, right? So the drones have, right? The drones don't have either fathers, they don't have father or son, but have grandfather and grandson. Right? right? So what does this mean here now? The drones, they do not have father or son, right? So they don't have a father or son, but they have a grandfather and a grandson. So how is that possible? Let us see here in the first case. This particular drone, we are saying that the drone does not have a son. Why a drone does not have a son? The drones undergo mitotic division to form the male gamete. Now this male gamete would fertilize with the female gamete to produce a diploid of female individual. So the drones will always have daughters, but not sons. Right. Now again, similarly, the drones will not have fathers. Why the drones will not have fathers? Let us take up this particular case. Right? Let us take up this particular case. This case, right? This is the second generation. So this would be the first generation. So this would be the second generation, and here this would be the third generation. So when we talk about the second generation individuals here, this drone do not have a father. Why they do not have a father? Because the drones are actually formed through a process called parthenogenesis from the queen. Right. So the queen undergoes meiotic division to form the haploid gametes, the female gametes. Now we said that this female gamete would now undergo a process called as parthenogenesis, that is organotomy, to produce the male drones. So males will have mothers, that is drones will have mothers, but not fathers. Similarly, the drones will have daughters, but not sons. Now what we are telling in the second statement here, they have grandfathers and grandsons. How do they have grandfathers and grandsons? Let us get back to the first generation. This drone in the second generation gave rise to a daughters, that is the female individual. Now this female individual again will start producing two different types of progeny. One is the female and the other one is male. So male which was seen in the first generation, male is absent in the second generation, and the same male is again present in the third generation. So drones will not have sons, but they have grandsons. Similarly, drones will not have fathers, but they will have grandfathers. Third generation again, right? So this drone here did not have father, it had only a mother, but it had grandfather. Right? So this is the thing what they usually say with regards to the honeybees here, where they say honeybees, in case of honeybees, the drones don't have father or son, but they have grandfather and grandson. Right? So this is the small topic what we have with regards to the haplodiploidy type of sex determination. Right? So we have taken up with three different types of sex determination, which are shown by different organisms. First one, chromosomal basis of sex determination. Second one was sex determination based on the X/A ratio of X chromosome to the autosome. And the third one is haplodiploidy type of sex determination. Right? So with this, we'll be done with our topic called as sex determination. Fine. Now, let's get into another topic here, which is called as mutation. Right? So we are done with sex determination. So the next topic what we'll be taking up is called as mutation. Right? I hope all are familiar with the term mutation because you would have already studied a few things with regards to mutation. So in this session and hopefully in the next session also, we'll be getting into a few details about the topic called as mutation. So we'll get into different types of mutations, what are those different types of mutation, and how the mutations are all being caused. Right? So the topic what we're going to take up is called as mutation. Right? Okay. So what exactly is this mutation? Right. So what exactly is this mutation? Mutation is nothing but any sudden change in the genetic makeup. Any sudden change in the genetic makeup of an organism or the genome of an organism, we start calling it as mutation. Right? Any sudden change. Right? Any change that takes place in the genetic makeup, we start calling it as mutation. Right? So the concept of mutation was first put forth or first discovered by a scientist by name Hugo de Vries. Right? So Hugo de Vries was the scientist who first came up with the concept called as mutation. So what exactly is mutation all about? Any sudden change in the genetic makeup, we call it as a mutation. So this concept of mutation was first put forth by a scientist by name Hugo de Vries while working on a plant called as evening prime rose. So while working on this plant called as evening prime rose, right? So Hugo de Vries came up with this concept. So evening prime rose, the common name, the scientific name of evening prime rose is Enothera lamarckiana. Enothera lamarckiana. So this is the scientific name of this particular organism, which is evening prime rose. So while working on this plant called as Enothera lamarckiana, which is evening prime rose, Hugo de Vries came up with the concept called as mutation. So he called mutation as any sudden change that occurs in the genetic makeup, right? He called it as mutation. So how exactly is this sudden change seen in the genetic makeup? So what could be the reason of this particular change in the genetic makeup? So the reason for the change in the genetic makeup would be because of certain agents which are called as mutagens or mutagens, right? So the agents which causes mutation, we call it as mutagens. So muta stands for mutation, gens stands for generating substances. So mutation generating substances or mutation generating agents, we start calling them as mutagens, right? So these mutagens would actually lead to some sort of changes in the genetic makeup. Right. So when we talk about mutagens, we have different types of mutagens. So all the mutagens could be classified into three different categories. Right. All the mutagens could be classified into three different categories. So what are those three different categories? The first one, we have something called as chemical mutagens. So this is the first category what we have. Chemical mutagens. Second one is called the physical mutagens. The physical mutagens. And the third one is called as biological mutagens. Right? So all the mutagens, that is mutation causing agents, would fall under these three categories, either chemical mutagens or physical mutagens or biological mutagens. So let us see what exactly are these chemical, physical and biological mutagens. So the first one what we'll be taking up is called the chemical mutagens. So when we talk about chemical mutagens, from the name itself we can make out there are certain chemical substances which are involved in bringing about the change in the genetic material. So if there is any chemical substances which are involved in bringing about the change in the genetics makeup, those chemical substances we start calling them as chemical mutagens. So these chemical mutagens could fall under different categories again. Right? So in turn we have classification for this. So it falls under different categories, of which the first one is called as base analogs. Base analogs. So this is the first category what we have under chemical mutagens. Base analogs. So what does this mean? Base analog? They are said to be analogous or very much similar to what? The base. Base here would be the nitrogen base. So any chemical substance which is very much similar to that of the nitrogen base, we put it under the category called as base analogs. So this is the first category what we have under chemical mutagens. So what exactly is a base analog? Any chemical substance which is similar to that of the nitrogen bases. So we start calling it as base analog. So what examples can we give here for base analog? The first one what we have is called 5-bromouracil. The first base analog would be called as 5-bromouracil. One of the examples of base analog, 5-bromouracil, we can call it as 5-BU, right? It is called as 5-bromouracil. So this is the first base analog. Similarly, we have one more base analog also, which is called 5-iodouracil. 5-iodouracil, which is called 5-IU, right? So 5-bromouracil as well as 5-iodouracil. So these compounds, they are said to be analogous to the nitrogen base called as thymine, right? So they are said to be analogous to thymine. So we have 5-bromouracil, 5-iodouracil, which falls under the category called as base analogs. The second category what we have under chemical mutagens is we call it as alkylating agents. Alkylating agents. So what does alkylating agents means? So they are the ones which actually adds an alkyl group. So therefore we call it as alkylating agents. So these alkylating agents also bring about some sort of changes in the genetic material. So what examples can we give here for alkylating agents? So the examples what we can use here is something called as mustard gas. I hope you would have heard about this gas. It is called the mustard gas, right? So mustard gas again would lead to some sort of changes in the genetic makeup, right? So this is one of the alkylating agents. One more example what we can give for alkylating agents is ethyl methane sulfonate. Ethyl methane sulfonate. In simple, we call it as EMS, right? Ethyl methane sulfonate or EMS. So these are the two examples what we can give for alkylating agents. So these alkylating agents also would lead to some sort of changes in the genetic makeup. The third category under chemical mutagens is we have something called as the acridine dyes. There are certain dyes which are present. So these dyes are also said to be chemical mutagens. So we put them under the category called as acridine dyes. So some examples what we can give for acridine dyes is we have a dye which is called as acridine orange. Acridine orange. Acridine orange is one dye, right? And one more dye. These are actually used in the laboratories, right, for doing certain experiments here. Then we have one more dye also, which is I think most familiar to you all, right? It is called as ethylium bromide. Ethylium bromide. We usually call it as ETBR, ethylium bromide, right? So this also falls under the category of acridine dyes. I hope you would have come across this ethylium bromide. So whenever we talk about this ethylium bromide, we say that it is said to be a carcinogen, right? So what does that mean, carcinogen? A cancer-causing substances. So how exactly cancer is caused? Again, due to some sort of sudden change in the genetic material, right? So we have ethylium bromide being a carcinogen, which falls under the category called as acridine dyes, right? So three different categories we have under chemical mutagens. First one is the base analogs. Second category, alkylating agents. The third category is acridine dyes. So a couple of examples in each of the case. So a couple of examples in case of base analog, we can give the example of 5-bromouracil and 5-iodouracil, right, which are said to be analogous to the nitrogen base called thymine. Then we have alkylating agents. So a few examples of alkylating agents. We have mustard gas as well as ethyl methane sulfonate, EMS. Then we have acridine dyes. So again, a couple of examples here. One is acridine orange and the other one is called the ethylium bromide. We usually use it in case of gel electrophoresis for staining the DNA, right? Ethylium bromide. So all these we put it under the chemical mutagen. So any of these chemicals would cause some sort of changes, sudden changes in the genetic makeup, leading to some sort of mutation in the particular individual. Right? So this is the first category what we have, which we call it as the chemical mutagens. Talking about the second one. Second one would be the physical mutagens. Right. So talking about the physical mutagens. So physical mutagens again could be of two different categories. Right. So physical mutagens also could be of two different categories. Right. These two categories we can call it as ionizing radiations. Ionizing radiations, and the other one would be non-ionizing radiations. Non-ionizing radiations. So these ionizing radiations and non-ionizing radiations both together fall under the category of physical mutagens. So what examples can we give here for ionizing radiations? We have the X-rays falling under this categories. Then we have the alpha rays. Then we have the beta rays. then we have the gamma rays. So all of them fall under the category of ionizing radiations, right? So they start ionizing the atoms that are present in the DNA, leading to some sort of changes in the genetic material, right? Okay, so we have the ionizing radiations, X-rays, alpha rays, beta rays, as well as gamma rays. Now, talking about non-ionizing radiations, we have one example here, which is UV rays, right? So UV rays falls under this category of non-ionizing radiation. Along with this, some sort of heat also would bring about some sort of changes in the genetic makeup, right? So these together, we start calling it as physical mutagens. So chemical mutagens, any sort of chemical substances, when it is seen on the DNA or when it affects the individual or the DNA of a particular organism, it brings about some sort of changes in the genetic makeup. Physical mutations also, same thing. We have ionizing radiations as well as the non-ionizing radiations, right? So the third category what we have here is called biological mutagens, right? So the third category would be biological mutagens. Under biological mutagens, we have certain viruses, right, which start acting as the biological mutagens, right? So viruses such as HIV, right? I hope all are familiar with HIV, right? Human immunovirus, right? AIDS causing virus. So this virus also would lead to some sort of changes in the genetic makeup. So we know how exactly HIV works, right? So whenever HIV particle enters into the body, what does it do? It starts integrating its genetic material into the human genetic material. So therefore, the human genome is being altered, bringing about some sort of mutation, right? So we have viruses acting on the acting as a biological vector. Then we have certain bacteria also, right? There are certain bacteria also. So these bacteria also would start bringing about some sort of damage to the DNA. It brings about DNA breaks, leading to damage in the DNA in case of humans. So those we classify them under biological vector, right? So any substance which causes mutation in a particular organism, we start calling it as a mutagen, right? So here we are talking about the mutagens, right? Mutation generating substances or mutation generating agents. So any substance or any agent which generates mutation, we start calling it as mutagen. Broadly, the mutagens could be classified under these three categories: chemical mutagens, physical mutagens, as well as the biological mutagens, right? So this is what we have with mutagens. Now, let's get into the classification of mutations, right? So let's get into the classification of mutation. So whenever we talk about classification of mutation, there are different ways how we start classifying mutations, right? So there are different ways on how we start classifying mutations. So we'll be taking up all of those different ways on how we actually classify mutations in this session and even in the later sessions also. So under that, let us first begin with the first type of classification. Classification based on the origin of mutation, right? So we are taking up with the first order classification, which is classification based on the origin of mutation. So on the basis of origin, we start classifying mutations into two categories, right? So we start classifying mutations into two categories. The first one, we call it as spontaneous mutations. We call it as spontaneous mutations. This is the first category what we have, spontaneous mutation. Second one would be induced mutations. Induced mutations, right? So the first category here is called as spontaneous mutation. The second one is termed as induced mutation. So what exactly is this spontaneous mutation? From the name itself, we can make out spontaneous is nothing but sudden. So if there is some sort of mutations which is happening suddenly by chance or in nature, such type of mutations we start calling as spontaneous mutation, right? So this type of spontaneous mutation occurs without the interference of the human activities or without the interference of humans. So if such sort of mutation occurs, that sort of mutation we start calling it as spontaneous mutation. right? So what exactly is spontaneous? Spontaneous means sudden. So suddenly, by chance, in nature, if there is a mutation happening, such sort of mutation we start calling as spontaneous mutation. Right. So this is the first category what we have. Second category is called induced mutation. So what is this induced mutation? From the name itself, again, we can make out induced. So we are inducing some sort of mutation. So wantedly, we are inducing some sort of mutation. So how are we inducing mutation? With the help of these mutagens. Right. So with the help of these mutagens, we are actually inducing some sort of mutations in the organism. So if you want to do any sort of work, any sort of research work, if you want to work on mutation, what we do, we usually induce mutation. So how do we induce mutation? Either by using a chemical agent, either by going in for using a base analog, or by using an alkylating agent, or by using the accretin test, or again by using ionizing radiations or non-ionizing radiation, or by using certain viruses or bacteria. So what we are actually doing, we are inducing mutations in the particular organism. Such type of mutation we start calling it as induced mutation, right? So this is usually taking place with human interference, whereas spontaneous mutation, it does not involve the interference of humans. It occurs by nature, it occurs by chance, and it occurs all of a sudden. Such type of mutations we start calling as spontaneous mutation as well as the induced mutation. So this is the first type of classification what we have, right? The first type of classification what we have, which is called as classification based on the origin, whether the mutation is said to be taking place spontaneously without the involvement of human activities, or it is taking place with the involvement of human activities. We call it as induced mutation, right? So this is what we have it. The first type of classification, how we actually classify mutations, right? So this is the first one, based on the origin of mutation, right? So spontaneous mutation as well as the induced mutation. So we have one more type of classification, what we actually call it as the major type of classification. So in this major type of classification, what we are going to do is, we are going to classify mutations again into two categories based on where exactly mutation is taking place. The first category was based on the origin of mutation. The second category what we are going to see is the major classification, where we are going to see the classification is actually based on where exactly mutations are happening. So based on where mutations are happening, we broadly classify mutations again into two categories. One we call it as chromosomal mutation, second one we call it as gene mutation. So if some sort of change in the genetic makeup occurs in the chromosome, such type of mutation we start calling it as chromosomal mutation. So if any sort of changes occurs in the gene of a particular chromosome, occurs in a gene present in a particular chromosome, such type of mutation we start calling it as gene mutation, right? So that mutation is what we actually call it as the major mutation, right? Major type of classification. So that major type of classification will actually complete it, or will actually look up to that classification in the next sessions, right? Thank you.